Okay, so I'm Benjamin Dunn. Thank you to the organizers for organizing this nice event. Um, as Vladimir said, I'm in Norway at NTNU, and I'm going to talk today about some work that we've been doing on looking at the state space of the activity of grid cells. Okay, so sorry, it's just okay. So the overall question that I want to talk about today is what is the underlying feature of the network activity of these grid cells? So I'll get into the details, but you can look at it in the context of the classical square environment versus an environment where grid cells can get screwed up, and then also in sleep. And these are all the lovely people that did the work, in particular Eric and Rich, who's over in the Moser lab with Edward and my Brett down the hill. So, okay, so grid cells are really fun neurons, and they're characterized by this very peculiar firing pattern that is spatial in nature. So up here in this video, you can see a rat running around as Dory or Jonathan Whitlock, either Dory Dirtickman or Jonathan Whitlock were throwing in crumbs, and the rat was running around, and then whenever the neuron fired, there was a dot placed in the location where the head was at that moment, right? Of course, after the fact. And at first you don't see much of a pattern, but as the animal just runs around the environment over, I think it's about 20 or 30 minutes, you see this beautiful pattern coming out, which is hexagonal in nature. And the cool thing is that if you look at multiple neurons, so down here on the bottom here are 5 or 6 neurons, each of which is recorded on a different tetrode, so it's probably not the same neuron. And each one has pretty much the same pattern in the sense that the fields are spaced the same way, and the distance between the fields are about the same. On the top is the spike map, and the bottom is the smooth drape map. And you can see the diameter of the fields and the spacing is just about the same in each of these neurons. But if you look carefully, you'll see that they're oriented, the orientation is also preserved, right? So relative to the wall, but they're shifted relative to each other. So if we're going to overlay these, you'd find that the fields are slightly shifted always a little bit to the left or the right or whatever for each neuron, suggesting that together they tile this space, all right? And this, of course, inspired many models. In particular, it inspired this notion that the underlying activity should live on some sort of toroidal manifold. So here is, again, an example of a single neuron. And as you would expect in an hexagonal pattern, the fields are repeating in 2 different directions. So one sort of left to right and the other one up and down, right? And if we cut out this little parallelogram and stick it over here, right? Again, we can see that in order for it to align, you actually have to connect around this way and then connect that this way, right? So if you follow those lines to apply the periodic boundaries, you get this nice torus, where here is one circle of the torus corresponding to one of these. You can't see my finger. That one. And then this one was the other one, right? So it gives you this nice little donut where the field now of that neuron is mapped onto that location of the torus, suggesting that the toroidal representation of that single neuron is right there. And if you looked at other neurons, you'd expect them to be shifted relative to this one on this torus. So this is sort of the background for a lot of modeling work and also a lot of our intuition of how grid cells work. But if you look in environments where you have grid cells that aren't square environments, so for example, here in the top left, you have this hairpin maze where the animals are just going back and forth all the time, then you don't see a very stereotypical, or you don't see a hexagonal pattern across the environment, right? It seems to be resetting at each time it goes around. Similarly, if you put the animal in a trapezoid, maybe in this large part of the environment, you see it more hexagonal, but then when it comes over here, it gets squished and kind of weird. I think these trapezoids are awesome, by the way. And then in the circular maze, you again have fields that are, they appear to be aligning with the pattern, but it's not obvious. In this square environment, this is a square environment that's separated by a wall. So it's actually two rectangular environments. And again, the fields are really squished. If you remove the wall, you see that the fields rearrange themselves and become a nice hexagonal pattern. But in this environment, it's squished. And even in big boxes, it starts distorting. So it really begs the question, what happens both in the wild and what is this pattern really? Is it just trying to maximally pack circles in there? Or what's going on? Right? What is the underlying activity? So what we chose to do is look at the activity of a large population of neurons in a box, and then compare the population activity in this maze here, this cross circle maze, and then also look at a non-spatial task, sleep, where there has been some indication that this representation is maintained, namely by these two papers here, Gardner and Toroidal, from last year, and but those were both done with pairwise cross correlation. So it suggested that yes, the torus should be maintained, but there's still room to explore there. So in terms of the data, we have data from these neural pixel probes, which are implanted in rats into the entorhinal cortex by Rich, who's up here in the corner. And these pixel probes are really, I think they're really changing the game in our field. I mean, we're recording for more neurons than ever, and we can do things that before we could only dream of. For example, in one of the rats, we have 124 grid cells recorded simultaneously from a single module. So you see all these neurons have pretty much the same spacing, orientation, just like those 6 that I mentioned before, only now it's 124. So it really opens the door to do things that we couldn't do before. And one of those things is, of course, look at a population analysis. So down here, we have this raster plot of the same neurons as above, and we can look at time points, so population vectors of this neural activity, and then project it down to a low dimensional space. For example, here on the bottom right, we see a projection into 3D using UMAP, which is a fun tool, right? And it is suggestive that we do have some sort of toroidal representation. It's more suggestive if I spin it around, but this isn't a video, so I can't do that. These are just two orientations of it. But you do see like a tiny hole here, and it kind of looks like a donut, right? But of course, UMAP is a visualization tool, and one of its drawbacks as a visualization tool is that we need our eyes. I mean, you could use UMAP in higher dimensions, but then you still can't see it. So in order to characterize this torus here, we're really looking and squinting and rotating. It's nicer to have a way to characterize these things without having to physically see it. So being able to see it in that higher dimensional space, right? Whether it's 5 or 100, but being able to look beyond 2 or 3 dimensions. And the way we've done this is going to the world of topological data analysis. In particular persistent cohomology, and sort of the building blocks of this framework are representing your data in terms of a simplicial complex. And what a simplicial complex is it's a structure that's made up of points, edges, triangular faces that you can build, and I'll show you how to build them in a second. But the 0 simplex is simply a point. 1 simplexes are the edges between those points. Then you can fill in the triangular faces, which gives you a 2 simplex, and so on, and put them all together, and you have a simplicial complex. And then once you have a simplicial complex, we can compute something known as homology. Right, and homology, I don't have time to explain it, but it essentially counts the number of holes in different dimensions. For example, here are 3 0-dimensional holes, so they're just connected components, right? There's 3 points, essentially. And these could be 3 like clusters or something, right? And then you'd have 3 connected components that each have no holes individually, they're just 0-dimensional holes. To the right of it, we have a nice circle, which is again represented by a single 0-dimensional hole and a single 1-dimensional hole, so the circle. Then we have a sphere, which is has a nice 2-dimensional hole, the interior of the sphere, and it's a single connected component. And then finally the torus, and then of course there's many shapes that you can characterize this way. And there we have 2 1-dimensional holes, the 2 axes of the torus, and a 2-dimensional hole in the middle where you would put the jelly in the donut, right? So the interior of the torus. And the way we can create these simplicial complexes so that we can then apply homology to is that one way of doing it, this is known as the Vittorius-Rips complex, is we take each point and we put an n-dimensional ball centered at each point. And then wherever they overlap, we create an edge, right? So between 2 points, we create an edge. And if they overlap across 3 points, each one is pairwise overlapped, then we fill that in with a triangular face and so on at the higher dimensions, right? And that gives us a simplicial complex for a given value of d, for a given diameter of our n-dimensional hole. But of course, then we have a hyperparameter, right? We have to find a value of d where that works for our data, which sounds terrible, but actually it's not, because what we can do is rather consider all values of d. So for example, here's a point cloud. And at each point, we're growing the ball, and we connect them, just as I said, right? And at some point, we connect them such that we actually get a hole in the middle, and the big hole. And eventually, when it's gone long enough, when the diameter of the hole is big enough, or the diameter of the balls are big enough, everything is connected. So again, as you're growing these balls, you get these circles, and you'll see one appearing here and one appearing there, right? But those don't live very long. So those holes are very short-lived, whereas this longer one actually lives quite a long time. And that's a notion that you have in this world of persistent homology, that the longer bars represent dominant features of your data and the shorter bars are noise. And there's sort of 2 standard ways of showing it. One is this barcode, and the other one is a persistence diagram, which again shows you the birth, like of the 2 small holes, and then it dies here, those 2 die here. And then the bigger hole is born later and then lasts much longer. So applying that to data, before, a few years ago, we applied it to head direction data because that's a very nice place to start. And also Adrian Peyrosh, when you're back in the Bujaki lab, made this data available on crcns.org. So it's a wonderful data set, very good for this kind of work. And we took these neurons, these head direction neurons, and of course, a single neuron will only tell you, like, when it's firing at a peak, you know you're maybe in this direction, right? This north by northeast. But as a population, you can get a much more refined representation of which angle you are, just intuitively, right? A population vector should tell you pretty well where you are in terms of direction, right? And head direction in the environment. So what you can do is take, again, the neural activity. Here we've smoothed it. And if you look in even just PCA space, I mean, this data is amazing. But even just in PCA space, you pretty much see a circle, right? But conveniently with this method, we don't have to rely on just the first 2 PCs. We can consider a higher dimensional space. And then when we look at there, we see a very nice one-dimensional hole, right? This single circle. And then using a method known as a circular parameterization, we can map this hole back to the unit circle. So it's nice because we can actually identify this specific 1D hole and then map it onto the coordinates of a unit circle. And then here I've color-coded, or Eric, this guy Eric Rebucken, who did the work, color-coded the points of the original point cloud according to the unit circle that was decoded. And on the right here, we had a video showing the animal's head direction from the tracking and head direction from the internal circle. So it was nice to see that they correspond. And there's some interesting details in that work, if you want to read it, as well as the other work that's related. And I also wanted to mention that, you know, this whole notion of applying persistent homology to neural data is actually a bit older. So going back to 2008 with Karina and Vladimir's work in this paper, and also Singh and Carlson back in 2008, and others as well. And so back to the grid cell data. What we were hoping to see is identification of a torus, right? And as we said, what we want to see from a torus is 2 one-dimensional holes and 1 two-dimensional hole where the jelly goes, right? And so we have this data, and we can take, for example, the data from this animal Roger, and look at the second module. Well, sorry, that's arbitrary, right? This just happens to be the second module. In this data set, we found 3 modules of grid cells. And if we take that population activity and use this method, we see one very long bar corresponding to the connected component, the 0-dimensional hole, 2 long bars here corresponding to the 1-dimensional holes. So these 2, well, I mean, 2 1-dimensional holes, and then one void, one 2-dimensional hole in this represented by this long bar, and lots of little stuff that we assume is noise, right? So from now on, the work you can assume is done by Eric Hermansen, which is this guy here, this very nice PhD student. So if we look for this in many of the data sets, so in all the data sets we find, or that we have, you see across different modules of different sizes, we see again this same, what they're called Betty numbers. So this 0-dimensional hole, 2 1-dimensional holes, and a 1 2-dimensional hole, like a void. In the square environments, as well as in the plus circle, in this cross circle maze, and the other ones as well, I just ran out of space. And so for one important thing is that just because we have the Betty numbers 1, 2, 1 doesn't mean we actually have a torus. You could have a different shape that also has the same Betty numbers. For example, a sphere with 2 circles, 2 1-dimensional holes attached to it. It would be weird, and I wouldn't know why we have that, but we could, theoretically. So one very nice thing that we can do is, like we did with the circle data, is we can take these 1-dimensional holes and map them onto a unit circle. So now we have a decoding of the position of the activity on each of these 1-dimensional holes. And remember, we think these correspond to these circles of the torus, right? So if we take those 2 1-dimensional holes and then map them back onto space, it should give us the 2 periods of the grid pattern. So here we see one that's going up and down, and then we see another one relative to it, right? So this is 60 degrees, more or less 60 degrees rotated relative to the first pattern, and also the spacing between these bumps. So this is basically a rate map of the decoded angle of the neural activity. So you look at the spacing here, it's 0.76 meters. So this suggests that we have a representation here by those 2 circles of this tile, which now looks very similar to that tile we talked about earlier, which is the repeating tile of the hexagonal pattern, right? Suggesting that not only do we have a torus, but we actually have an hexagonal torus. And if we do this for all the different modules that we have, we can see that they line up quite nicely, and they're growing in size, more or less the square root of 2, but not exactly. And then also in the different conditions. So here's the red and the blue are the open field in this plus circle maze. So another thing we can do with the decoding is look at it, right? We can look at it as the animal is running through the environment. So here we have the animal going through space. So you see his real trajectory. And at the same time, you can look at the activity across the toroidal mapping, right? And you can see that as the animal is going left, it goes left. As it goes down, it goes down. And it's repeating because, well, now the video is repeating because it's on a loop, but the activity is repeating over the edge of the torus back and mapped onto the opposite side because it's a repeating tile, right? It's the toroidal representation of the activity. The nice thing is that we see a good mapping between the decoded trajectory on the torus and the trajectory through physical space of the animal. And of course, looking at this mapping, you say, well, they're not exactly the same. They're close, but they differ occasionally, right? And are those differences significant? Meaning, does one explain the neural data better than the other? So we looked at it 3 different ways, in terms of trying to compare which one is better. So again, you can look at the activity of the single neurons as, here's physical space, and here's the toroidal mapping. You get a single field in that toroidal space. And so now we can calculate things like the spatial information, by the Skaggs-McNaughton approximate mutual information, and compare the scores you get from it. And that's what you see up here. And here the toroidal representation explains more, both in the open field and the funky maze. We can also look at the log likelihood difference in terms of which one is explaining the single neuron activity better in a GLM. And all of this is done on held out data, so not the data that we fit to. And then in this case as well, looking at the log likelihood difference, the torus wins as well. Or we can look at covariance matrices, so the covariance between the activity of the single neurons. And this is just the raw covariance matrix. We can look at what you would predict from the toroidal representation on held out data, and then look at the residuals, and you see it captures most of the variance in the data. And I didn't show it here, but the torus explains or captures more than the space. So these three things together suggest that the torus is actually a better description of what a grid cell is doing than actually saying it's spatial, right? I mean, it's spatial in the sense that these things are mapped on, that the torus is mapped onto space, but the torus actually explains more, explains the data better. Okay, here, there we go. I'm gonna make sure I didn't skip a slide. Okay. So then we wanted to see if the mapping is preserved across environments. So again, we have these two environments. We have the open field and the circular track. Open field gives you a beautiful autocorrelation, as you'd expect. Circular track gives us not such a beautiful one. I mean, you can still kind of see something, but you know, it's not so obvious. If you look at this, you wouldn't say, aha, grid cell, you know, and think maybe it's something. But in the toroidal representation, it's preserved, right? So you have the activity of the field in the same location each time. And if you look across all the neurons in this case of this module, you can see, I know this is small and a little pixelated, but you see that the tuning fields are always in the same place for the same neuron. And I should say, one thing we did do is once you have the decoding, of course, the angles are relative, or they're just, you know, they are what they are. So you rotate as a population such that they match. But it's the whole population is rotated together. So here you see a nice column of, looks like the same neuron repeated, but it's not, I promise. They're different neurons. And they actually cover pretty well the phase space. For example, here you can see this is the single tile, and all the green dots are dots from the open field, or sorry, the circular track. This is the center of the field of all those guys, and you see it covers this phase space pretty well. And importantly, that location and phase space is maintained in the new environment, or so open field and circular track. And the blue lines represent the distance between peaks of the same neuron, so the center of the field of the same neuron. So it's really covering most of the phase space, maybe a little clumping here and there, but not much. And it the distances are preserved. And this you can see in these cumulative distribution functions here for the correlation between the rate maps, these toroidal rate maps, where the data really it hugs one versus a shuffled distribution, which is just doesn't hug one at all. And similarly, the distance between the peaks, which is very close to zero in the data, right? So strongly suggesting that this toroidal representation is maintained across the two environments. We do the same thing with sleep. So we have REM sleep and slow wave sleep. These are, in this case, is the same module, this Roger, second module of Roger. And we see again these strong one-dimensional holes and that strong two-dimensional hole that corresponds to, or that we hope corresponds to a torus. And then we can do the decoding. And again, you see these fields just align nicely in each of the three conditions, open field and slow wave sleep and REM. Of course, we don't have the spatial maps for the sleep because the animals are asleep, they're not moving. So those are very uninteresting. And you see that they correspond both visually, and you can see it in the phase space mapping and the statistics of that. And I just wanted to throw in a whole bunch of them so you can see that it's very remarkable. And when we have this is just one module. We have a bunch of them. It's very fun to watch. So as a summary, we have cool. We have the characterize the state space, or the a notion of the shape of the state space of a grid cell module, and we found it to be toroidal, hexagonal torus. And we find that this is maintained across environments and during non-spatial tasks, such as REM and slow wave sleep. And we find that the torus actually explains the neural activity better than physical space, which was a bit of a surprise. I was actually expecting physical space to be a little bit better. And something I didn't show because I didn't have time, but this of course fits very well with the predictions of it fits very well with the modeling work of the continuous attractor model. So if you generate data from different attractor models, you see the same thing. And of course that framework, you have a fixed connectivity, so it predicts that you will always maintain the activity on the torus. So it's very cool. So again, here's all the people who did the work, mainly Eric and Rich. And then Eric Rebucken, he did the head direction stuff. These are funding agencies. And just a quick plug, we have an open postdoc position starting soon. Norway is a great place to live, especially during pandemics. And the social system's amazing, and it's a good place for science. We have, Nobel Prize winners and stuff down the hall. And it's a great place. So anyone interested, please email me. It's a 3 or 4 year position, depending on if you want to teach or not.
Okay,
so, there we go.
Thank you for your wonderful talk. So now is the time for questions. So the first question is by Cosio, and the question reads as follows: how do you determine which components in the birth-depth diagrams are noise? Couldn't a short component also be due to more fine-grained but stable structure of the manifold?
Yeah, so that's a good question. So what we did here, which is kind of a hack, it's not an established way. I mean, this is a general problem within the world of persistent homology, like what is noise and what isn't noise. It could be interesting even though it's not long, right? What we did here sort of as a second check, which is a standard hack in neuroscience, is that you shuffle things. So you take each activity of each neuron and you shuffle it randomly a whole bunch of times. And then each time you have, you've rolled the neural activity here and there, you go through the whole method again and you count the number of holes. And that gives you some notion of what you'd expect by chance. And that's what these orange bars here is, that it's what we think is the chance level for these features to appear. Yeah.
Okay, and we have time for just one more question. So another question is from Tom Burns. Well, actually it's 3 questions, but okay, the first question: what is surface topology? What surface topology would you expect for grid cells in 3D G beds?
Ooh, so it would be really cool if it was a higher dimensional torus, especially because that would totally justify this method, because you could never see that with the UMAP or something in 3D. I don't know. I honestly don't know. I've looked at the data. I mean, and just in the paper, Nachum gives great talks. And so I guess a torus, but I honestly don't know. Sorry.
And the last question is philosophical. Maybe since we have short on time, you can answer it briefly. Okay, topological data analysis seem to always suggest low dimensionality represented by lots of neurons. If true, why did evolution give us so many neurons to encode something so low dimensional?
Ooh, redundancy.
Very good. That's perfect answer. So unfortunately, we have to stop. And other questions perhaps can be handled online.